Hello, student. I want to welcome you to the world of biology. My name is Professor Sami M. Ayodele from the Department of Biology, Faculty of Science, National Open University of Nigeria. My welcome address is on the course page, which I advise that you should go through. Always visit the course page of this course. I'm going to handle BIO192 Practical Biology 2 for this facilitation period. So I want to start with the first week lesson, which has to do with the bacterial kingdoms. And we have some learning outcome that we want you to really understand at the end of this lesson 1. So at the end of this class, we expect that you should be able to state the diagnostic features of the kingdom bacteria. Then you should be able to identify the major phyla of Archaebacteria. Then being able to differentiate the 3 groups of Archaebacteria. And then know the observable adaptive features of the Archaebacteria. So that is, you should be able to know what are the kingdoms in which the bacteria belong. How do you identify this kingdom or the phyla under the kingdom? Then, what are the group of Archaebacteria, and also what are some of the features that enable them to survive in their environment? For introduction, so this group of organisms is one of the 6 kingdoms, that is Archaebacteria is one of the 6 kingdom classification of living things. As we know, the 6 classification include the plant kingdom, the animal kingdom, the fungi kingdom, then we have the protista, and then we have the monera, and then we have the bacteria. So then the word archaea, what it means is that it is ancient, that is very old, primitive. And this kingdom contains organisms at the simplest level of organization. They are mostly unicellular. They are very primitive in nature. So the Archaebacteria simply mean ancient bacteria, that is old bacteria. And in fact, some people even postulate that probably the life originate from the bacteria. So, and they are believed to be one of the first group of living things to occupy the surface of the earth. So they are prokaryotes, or we can say they are prokaryotic, and they have been found in habitats otherwise thought of are inhabitable. That is, they are found in some environment that we think that no living thing can survive under such an environment. So the members are prokaryotic, as I've said earlier on, and they were formerly classified as member of Monera. So, but because of some structural feature that different from that of the Monera, they now form their own domain, and that's why we place them on their own kingdom. So, an archaea do not contain peptidoglycan in their cell wall, as we found in the cell wall of other bacteria. So they are the only group of organisms with lipid in their cell membrane. So other living organisms do not have lipid in their cell membrane, but this group, Archaebacteria, have lipid in their cell membrane. So archaea also are single cell organisms that can survive without oxygen. They are unicellular, and they can be found where there's no presence of oxygen. So, and they can withstand high temperature as high as 100 over 100 degrees centigrade. So they are found in the extreme temperature, and they can survive under that extreme temperature. So the structure and function of their genes is similar to that of eukaryotes or eukaryotic organisms. So what are the features now that distinguish archaebacteria? That is the diagnostic features of archaebacteria. So we have some of these features that distinguish them. One, no peptidoglycan in their cell wall. They lack this peptidoglycan in their cell wall. Two, the cell wall is made up of glycoprotein and polysaccharide. That is what is the nature of the cell wall, made up of glycoprotein and polysaccharide. So three, the cell wall envelopes have a high resistance to antibiotics and lytic agents. So it is because of the nature of the cell wall that makes some of them to be resistant to antibiotic. So they have a very different lipid bilayer making up the cell membrane. So in the cell membrane, you have a high bilipid layers that make up the cell membrane. 5. The RNA polymerase of archaea is very similar to that of eukaryotes or eukaryotic organism. So they have a very similar RNA polymerase, which is an enzyme. 6. The eukaryote and archaea ribosomal protein are similar to each other. So the ribosomal protein are very similar to that of eukaryotic organisms. Then archaeobacteria are about 1/10 of a micrometer, and then also it will extend to about 15 micrometer in size. And some of them are flagellated. Some of them have flagella, which is different from the flagella of other bacteria. And then this flagella is an organ of locomotion in this group of bacteria. Number 9, the archaeobacteria also are not pathogenic bacteria that live in and around other organisms. So when we say non-pathogenic, they are not capable of causing diseases. Excuse me. So they are not capable of causing diseases in both plants as well as animals. They are autotrophs. That is, they are capable of, some of them are capable of manufacturing their own food. So that is autotrophic mode of nutrition. And 11, they use carbon dioxide in the atmosphere as a source of carbon for the process called carbon fixation. And carbon fixation takes place during photosynthesis. And that's why they are autotroph. We say they are autotroph. They are able to fix carbon dioxide to form a chemical energy through the process called photosynthesis. Then how do we classify archaebacteria? Their classification, grouping them. So, and we classify them into 4 phyla. And this classification is based on certain criteria. So we are going to see 4 phyla of archaebacteria, and we give a typical example of each phylum. One of the phylum or the phyla is Urocheiota, and a typical example of it is Pyrococcus abscess. Then the second phylum is Crinacheiota, and a typical example is Pseudophilobus acidocandarisu. So the third one is Nanocheiota, and then Nanochium equitans is an example of this phylum. Then we have Coracoeta, and a typical example of this phylum is Candidatus coraceum. So these are the 4 basic phyla that we have in archaebacteria. Then the structure, how do they look like? You can see the structure. When you observe it under the microscope, this is what you see as a typical structure of archaebacteria, that is the morphology, the appearance of archaebacteria. So as I've talked about the size, the diameter of this group of bacteria cell range from about 0.002 to 0.004. So that is 0.5 to 1.0 micrometer. So, and then the volume of their cells is only around 1,000 that of a typical eukaryotic cell. Eukaryotic cell is an advanced cell, so which are more advanced than that of these archaeobacterial cells. So, and then we have three forms or shape that are found in archaeobacteria. So they have different shape. About three types of shape are recognized in archaeobacteria. And then the three shapes found in archaeobacteria are spherical cells that are called cocci. We have those that look like rod, and we say rod-shaped cells. And then these are called the bacilli. Then the third type of shape is spiral-shaped cells, which can either be fibrous, spirillum, or spirochete. And some other shapes may also occur, the different forms. Some may be regular, some may be rectangular, some could even be square. So some could be in form of filament, some could aggregate together to form a colony. Then what are their structures? The structure of an archaeobacteria. So the organism have no membrane-bound organelles, just like we have in some prokaryotic, and like other prokaryotics. As we know, we said that prokaryotic are primitive organisms. They do not have organelles like that of eukaryotic organisms. So, and even the structure they have in their cell are non-membrane bound, as we found in the case of advanced cell like eukaryotic cells. So they lack some organelles. Some of the organelles that are not found in them, that are found in eukaryotic cells, include nuclei. They do not have nuclei. They do not have mitochondria. They do not have endoplasmic reticulum. So they have no Golgi complexes or even chloroplast in them. So the cell contains a thick cytoplasm that contain all of the molecules that are found in them, particularly for metabolism and nutrition. Then also, their cell wall, as we have said earlier on, do not contain peptidoglycan that is found in other cells. So, and the group have a very rigid cell wall, and it is this rigid cell wall that supports the cell. It is the rigid cell wall also that allow an ecobacterium to maintain its shape, the structure. It is also the same rigid cell wall that protect the cell from bursting, particularly when they find themselves in a high salty environment, that is hypotonic environment. So, and because of this lack of nucleus, the genetic materials of this archaeobacteria spread in the cytoplasm. So they float freely in their cytoplasm. Their DNA is a single circular molecule, single-stranded DNA molecule that is found in their cell. Then there's little or no protein that is associated with the DNA in the cell. So then, but there are plasmids that are very small. The plasmids are circular pieces of DNA, and then this can duplicate independent of the larger genomic DNA cycle in other organisms. So it is plasmid that code for particular enzymes or for antibiotic resistance in the organisms. Where are they found? That is the environment where they live, the habitat. Where are archaeobacteria commonly found, or where do they live? So archaeobacteria can be found in an environment that is high in temperature. Even I've said it, in temperature above 100 degrees centigrade. So, and then they can also be found where there's no oxygen and light. So they are anaerobic. Anaerobic organisms are organisms that do not require oxygen, either for respiration or to carry out their activities. So they are known as ectomorphites. That is, they are found in an environment that we think that any organism cannot survive. So they can be found in the following habitats. That is where they are commonly found. near volcanic activities, and of course, where there's volcanic eruption, the temperature there is always very high and terrible, very serious. So then they can be found in geysers, then black smokers and oil wells. Archaebacteria can be found. In fact, some of them are even used to detect the presence of oil. Then they can be found in deep oceans. And of course, when we talk about ocean, that is a salty environment, so they can live in salty habitat. Then they can be found in very cold habitat in Arctic, in the polar region. They can live successfully there because we said they can survive at extreme conditions. So then they can also be found in highly saline, acidic, or alkaline water, both alkaline, whether the environment is acidic or it is contain a lot of saline. The Archaebacteria can survive. You can find them there. Then in the marshland, that is swampy environment, so they survive. They found them there. And they live also in sewage, so they live in the soil, and then they can be found in the gut of humans and ruminants, so in the gut of animals. And then what are their groups? The different groups of Archaebacteria. So, and with this grouping is based on the carbon dioxide fixation. So we said that some of them are autotrophs, that they use carbon dioxide in the atmosphere. And in fact, they help in the reduction of the amount of carbon dioxide, which is causing what we call ozone layer. So they can fix carbon dioxide in the atmosphere, and the process is called carbon fixation. And then for them to survive in their harsh environment also, they employ several chemical reactions to enable them survive where they live. So, and on the basis of the nature of their habitat, we also group them into 3 different groups. So, and then the 3 groups that we group them on the basis of the type of environment include methanogens, the halophiles, the thermoacidophiles. So, and then the methanogens, they are those that can reduce carbon dioxide into methane, and then particularly in the clean environment, in the marshy, swampy environment. So they carry out this activity. So they are found in this environment. So then this group can only survive in the absence of oxygen. And that's why if you go to a stagnant water where there's bubbles, it means that a decaying process. So there's lack of oxygen there. So, and what is bringing about this decaying process are the bacteria. So they produce mass gas that one can observe as a bubble in stagnant waters. The gas that they produce could be offensive, it may not. So depending on the process that is involved in the production of the gas. So they are also present in the gut of cattle and termites. You find them, and probably they play some role, maybe in the digestive system of these organisms or animals. The bacteria that are rod-shaped or those that are spherical can be gram-positive as well as negative. So we will talk about these 2 groups basically. So the gram-positive and gram-negative bacteria. So, and we have some typical examples, which include methanobacterium and then methanococcus and some other examples. Then we have the halophiles. And these halophiles, they are found in areas with very high salt concentration, such as in the sea. As I've said earlier on, that sea is a saltwater environment. So, and then those bacteria that are found there are called the halophiles. So they contain vitrophodopicins, so which is a red or orange pigment in them, which enable them to survive under these conditions. So we have some examples also, which include halobacterium, Halococcus, Natronabacterium. These are examples of the halophiles that are found in salty environment. Then we have the thermoacidophiles, and these are the group of bacteria that can survive in an extremely high temperature. Excuse me. So they are also found in low pH level, that is in the alkaline, could be in the also an acidic environment. So, and they can survive at over 100 degrees Celsius with a very low pH of 2. So, and most of these organisms are anaerobic in nature, that is, they can carry out respiration in the absence of oxygen. So, we have some of the example of these thermoacidophiles as pyrodictum, we have pyrococcus, and then we have Staphylococcus and Thermococcus, and so on. Then what are their adaptive features? That is, what are the features that enable them to live successfully in the environment? So what makes them, or what are the structure they have that make them to survive in the environment? So the archaebacteria do not form spores like some other bacteria that are known, that under certain conditions, so some of them will produce spores. So, but a few species of haloarchae undergoes phenotypic switching. That is, they can change their physical structures depending on the conditions of the environment. So, and that is they can grow several different cell types that are resistant to osmotic shock. So it means they can change their structures depending on the environment or environmental conditions that is prevailing at that particular time. So, and then this means the organism can survive in aquatic environment with low salt concentration. Even those in the fresh water that lack oxygen, some of them also can live in that type of environment. So now, having seen the archaebacteria, which belong to the phylum of bacteria, so now we need to look at some of the activities. In your course materials, if you look at under this topic 1, so you will see that there are some structures. So that is the one I show you as part of their structures. So then, if you look at them critically, then what are some of the features of the archaea that you can see in them? In the structure that I show you in the slide, in the past slide, in two structures, that when you observe them under the microscope, that's what you will see. So what are the features that you can see in them? Then I will advise you also, for practical purposes, you visit a stagnant water site, especially in swampy environment, particularly if you are living in the Niger Delta. There are some swampy environment that when you go to visit such environment, you will see that there are bubbles that will be coming up. So then, what group of organisms do you think are responsible for such bubbles that we observe in this marshy and swampy environment? So of course, we know, we have talked about them. So basically, what are they? So then, so now we talk about the other kingdom that we have under the bacteria. And this kingdom is Eubacteria. In fact, this group of Eubacteria, they are the one we refer to as true bacteria. So the archaebacteria, we say they are primitive, they are ancient, but we refer to these Eubacteria as the true bacteria. And they are the second kingdom in the 6 kingdom classification of living things that I've just mentioned at the beginning of this class. So they are prokaryote also, just like archaebacteria, and they are found everywhere. There's nowhere you do not find bacteria. There's nowhere in the whole world you do not find bacteria. So, and I always tell students that the number of bacteria in the entire universe, if you pack them together, it supersedes the weight of the entire bacteria in the universe, supersedes all other weights of other organisms combined together. And that's how to tell you how numerous are they, and they are found everywhere. As we have said, no matter how terrible the condition is, you will find bacteria. So they can be found in the bodies of plants and animals. Then they cannot be seen with ordinary eyes. Our naked eyes cannot see bacteria. It is because they are so tiny, they are very microscopic. We only study them under the microscope. And a lot of them live as single cells. They are not multicellular cells, they are single cells, majority, if not all. So except those that will form structures, but they are of individual cells. So they live as single cells. So, but they are able to produce colonies, link up in chain to form filaments, and then they will appear like a multicellular organism. But they are individual cells that form such a filament or colonies. Where do we find bacteria too? Just as the places where you can find archaebacteria. So, and they, as we have said, that they are found everywhere. They are cosmopolitan in nature, and then they live everywhere, no matter how terrible the environment is. Bacteria can live there. So they can be found in soil, find them in water, find them in feces, find them in decaying substances, also in the body of plant. Every human being carries bacteria. The luck we have is that it's not all bacteria that are pathogenic, not all of them that can cause diseases. So, but we carry bacteria here and there, anywhere we go, anywhere we find ourselves, we carry bacteria. Then, how do we classify them? How do we classify bacteria? So, the bacteria are classified based on three criteria. We use three criteria to classify them. And then these three criteria include: one, shape, the structure, the way they look like. It is easy to distinguish the bacteria based on their shape. So, and on the basis of the shape, we have three main shapes, as you can see on the right hand of this slide. That is the bacterial shape. We have those that are spirular, we have those that are bacilli, and we have those that are cocci. So the cocci one are the ones that are round in shape. The bacilli, they are the ones that look like rod, and then the spirular are the ones that look like comma, and as you can see, like a spring. So these are the three basic shapes that are recognized in bacteria. So they can be classified also based on their nature of growth, that is their growth characteristic patterns. So, and on the basis of that one, those that appear in pairs, we refer to them as diplo. We use a prefix called diplo, either diplococcus. Then those that combine together and form a shape like a grapes or football, so we use a prefix like a staphylo, meaning that the bacterial cells are arranged in clusters, and then we can say staphylococcus or staphylococci in plural. So also we use the prefix strepto, and this means that the bacteria are arranged in chain, and we refer to such a bacterial group as a streptococci or streptococcus. Then also we use the type of cell wall structure to group them. So, and on the basis of this, so the cell structure will depend on the reaction of the cell wall to staining techniques. We use a staining technique that is called gram staining to group bacteria into two basic groups. So, and then based on gram staining, the cell wall can be classified into two: gram positive, which are gram positive are bacteria that have simple cell wall. That is, their cell wall can easily be stained. So, and then the cell wall are made up of only one layer of peptidoglycan before the plasma, as you can see. So, when stained with violet and red dye, gram-positive bacteria appear purple. That is when you stain it with gram stain. So, and that's why we refer to it. It retain the stain. So, and it will appear purple, and we refer to it as gram-positive bacteria. Sometimes blue, it could be blue, it depends on the temperature of the environment. Then the second group are the gram-negative. And the gram-negative bacteria, they have more complex cell wall. So they do not retain the stain. So, and the cell consisting of one layer of a lipopolysaccharide membrane and a peptidoglycan layer. So they have a more complex cell wall than the gram-positive. So, and the gram-negative bacteria usually appear red when you stain it with violet and red dye. So they appear red. So it is that of the gram-positive that appear purple when you stain them with violet and red dye. Then also we group them on the basis of carbon fixation or the carbon source. So, and then based on these carbon and energy sources, we can classify them into photoautotrophs, that is those that can use the sun energy to carry out their nutrition. Then we have photoheterotrophics, those that can use sun energy but they still depend on the other food that are manufactured. Then we have the autotrophic bacteria, that is those that contain the pigment that enable them to fix carbon dioxide, then using the sun energy. Then we have heterotrophic bacteria, that is those that cannot fix carbon dioxide, they cannot manufacture their own food through the process of photosynthesis or chemosynthesis. Then we talk about the structure of bacteria. So as a prokaryote, they do not have nuclear membrane. There are no nuclear membrane. So they have prokaryotic chromosomes. Remember, we said the chromosomes are not embedded in a clear nucleus as we have in eukaryotic cells. So this genetic material, we said they are scattered in their cytoplasm. So, and some bacteria have specialized internal membrane. We have example as cyanobacteria. Cyanobacteria, we before call them blue-green algae, is what we call them. They are the one that we refer to as a cyanobacteria. So, and they have a membrane that contains chlorophyll and other chemicals that are used to carry out photosynthesis. So, because of the presence of this chlorophyll, they refer to them as autotrophs too, because they are capable of carrying out photosynthesis. We have some bacteria also that have cell walls similar to that of plant cell. So, but in that case, they do not contain cellulose as we have in plant cell. So, but their cell wall are made up of peptidoglycan that is present in the cell wall, unlike in the case of plant cell that contain cellulose. So, in some members, there is an outer layer that we call the outer membrane. And even some even have what we call capsule. So this is a coating layer on the wall of their cell. So, and we refer to it as capsule. And this capsule also plays some role in protection of some bacteria. So the coating is made up of complex sugars and serve to protect the cell against environmental dangers. So even including the enzymes of the host where they find themselves, for those that are parasitic, so or those that are pathogenic. So, and some of the dangers also include attack by host immune defenses or dehydration, that is a reduction in the amount of water in their system. So it is this capsule or the coating that prevent this dehydration. Then what are also some of the features that enable them to live successfully in the environment or in their habitat? So we have some features that enable bacteria to live effectively in their habitat. And some of these features include the shape. For example, we have spiral shape. And this spiral shape enable them to move through fluid more easily than can cocci or bacilliar bacteria will move. So this spiral shape gives them an advantage of their movement in the fluid. Then also the presence of plasmid in them also enable them to live successfully in the environment, and this can be transmitted from one cell to another. So the presence of this the plasmid can be transmitted from one cell to another cell. Then the ability to trade genes with one another makes bacteria amazingly adaptable to the environment. So it means they can exchange genes. The genes can be exchanged from one bacterium to another. So it is one of the things that enable them to even adapt successfully to their environment. So the presence of beneficial genes also in them, like those for antibiotic resistance, may be spread very rapidly through bacterial population. So since they are able to exchange genes, so it is possible for them, this what we call beneficial gene can be also transmitted from one bacterium to another. Then the presence of capsule, which we had already mentioned also earlier on, so serve as protection, and it protects the cell against environmental danger excuse me, such as attack by the host immune defenses, as we have said earlier on also, and it prevents the dehydration of the bacterial cell. The fourth features is the presence of endospores. So, and these endospores are formed when surrounding conditions are not conducive for them. So it is then now the result of the formation of endospores, so that they will be able to live successfully in the harsh environment. So, and these endospores have what we call thick wall for protection. So it is this thick wall that protect them from the danger of the environment or the harsh environmental condition. And then another feature is the outer membrane. And then this outer membrane increases the potential surface area for their photosynthesis, particularly those that are photosynthetic. So now we haven't talked about bacteria. We have looked at the phylum, bacteria, archaebacteria. We have looked at the activities about the archaebacteria. So we are going to look at some practical activities about this bacteria, that is staining method in bacteria. And as we have said, that we use staining techniques to group bacteria into two major groups: gram positive and gram negative. So we are going to look at the procedure involved in these staining techniques or staining methods. So one, and then to stain bacteria for examination under the microscope. So what we do is that there is a method that are used in this staining technique in order to identify the bacteria or group them. So, and then the staining method separates nearly all bacteria into the two major groups, that either gram positive or gram negative. So, and it is this staining is based on whether or not they resist decolorization of methyl violent and subsequent treatment with iodine. So then what are the procedures in this staining technique we are talking about? So, and first, we are to make a smear of the bacterial culture that may be provided. And then this smear will be done on a clean and grease-free slides, microscope slides. And then after the smear, then we air dry the film by waving it. You just wave it. When you have made the smear on the slide, you just wave it so that you can air dry it. Then after that, you will now fix the smear by waving it over a Bunsen flame or Bunsen burner. So this will be demonstrated during the practical class, physical practical class for you. So then, having fix it on the Bunsen flame or Bunsen burner, then place the slide on the rack over a sink of water. And then what you do is that you cover the smear with crystal violent reagent for about 1 minute. And then when you have covered it with a crystal reagent, then you will rinse the slide in the slowly running tap for about 5 seconds. So that you can remove the excess stain. Then rinse the slide with grams iodine and flood it with the same reagent for 1 minute also. Then you rinse again in a slowly running tap. And after that, you apply alcohol reagent slowly until no more dye runs off from the smear. So that is probably most of the dye might have been removed from the running tap water. Then cover smear with safranin reagent for about 30 seconds. And then you rinse slide under slowly running water again. Then you blot the dye using paper towel. And then please, when you are blotting, don't rub it. Just press it and remove the paper. If you scrub it or you rub it, then it will remove your specimen. Then observe the slide using the oil immersion lens of the microscope. And the oil immersion lens of the microscope is times 40, so that you can observe clearly the bacteria. Whether on that, you can even identify the shape, and then you can also group them into whether they are gram positive or gram negative. So, and after these processes, what you need to do, you record your observations, and your observation could be the grouping into gram positive or gram negative, or the shape, the different shape of the bacteria that you see under the microscope. So then you can now state what are the differences between the gram positive and gram negative bacteria. So then we have talked about some of these differences in the process of the discussion. Then how to observe the shape of bacteria under the microscope also. We have some procedure for that. And one is that you use a sterile loop, and use this sterile loop to take a small portion of the bacteria colonies from your culture, or the culture that may be provided for you during the practical exercise. And then what you do is that you smear this evenly, you spread it evenly on the microscope slides. And then you fix it over a flame, Bunsen burner or Bunsen flame. So by passing the under surface of the slide swiftly through the top of the flame. And then you flood the dry smear with either methylene blue or crystal fouling and leave it for about 3 minutes. And then after that 3 minutes, you drain dry again. And then you lower a cover slip over the preparation and observe your material under the microscope. And then you will see the cultured bacteria. Then you'll be able to see the different shape. Then you'll be able to group them or classify them into either coccus, bacillus, or streptococcus, or bacillus, or rod shape, cocci, and so on. So after this procedure, you'll be able to identify the different shapes